Okay, we're going to shift now to the presentation for our first speaker. Dr. Tracy Leskey received her PhD in entomology from the University of Massachusetts at Amherst. She has been employed as a research entomologist for USDA ARS at the Appalachian Fruit Research Station in Kearneysville, West Virginia since 2010. Her research focuses on behavioral and chemical ecology of economically important tree fruit pests, and specifically on developing behaviorally based monitoring tools and management strategies for the brown marmorated stinkbug, plum curculio, apple maggot fly, and dogwood borer. Tracy?
Yes, can you hear me, Kathy?
Yes.
Okay, I'll go ahead then. Yes, so today I am going to talk about brown marmorated stinkbug and its emergence as a serious pest of agriculture here in the Mid-Atlantic. To begin with, brown marmorated stinkbug is considered an invasive species, and it is native to China, Japan, Korea, and Taiwan. It's defined as an invasive species because it has been introduced into an area for which it naturally does not occur, in this case North America, and also because it threatens both environmental and economic resources due to the damage it causes, in this case to agricultural crops. Now, a little bit about the identification of brown marmorated stinkbug if you haven't seen these insects yet. The adults you'll see on the right-hand side of the screen, the lower right, with the male and the female. They have very prominent white stripes on their antennae. You can see the small arrow that's pointing at the left antenna of the male stinkbug. On the outer edges of the abdomen, there are alternating patterns of white and dark markings, and this is actually why it is referred to as the marmorated stinkbug. It's another term for describing that appearance. The underside is generally pale with some gray or black markings, and of course they emit a very pungent odor when disturbed, hence they're called stink bugs. And I can tell you, if you haven't smelled it previously, it smells very different than our native species, and some people often detect an odor of cilantro when they handle this bug, although it is much more pungent than just the odor of cilantro. Now, the eggs in the upper right-hand corner are barrel-shaped, and when they're first laid, they're kind of bright green, but they turn white. In general, they're laid on the undersides of leaves in masses, and there are typically 28 eggs per mass. The female lays eggs in intervals of 7, typically. The nymphs, you see the first instar nymphs there just below the eggs. The nymphs, typically, the first instars will be near the hatched eggs and they will remain there. The seconds through the fifths are very mobile, and in fact they've been described as sort of tick-like when they're climbing around on foliage. But you can see that they typically also have those white stripes on the antennae, and again, that's a good way to define if these are brown marmorated stinkbug nymphs. Now, a little bit about the history of brown marmorated stinkbug, and these comments are related principally to tree fruit because this is where we've seen the most injury, at least that we're most familiar with. This insect is thought to have been introduced into the United States somewhere between the mid to late 1990s. It showed up in Allentown, Pennsylvania in 1998, but the first properly identified specimen didn't occur until 2001 when specimens were sent by Karen Bernhard, who is with Penn State Cooperative Extension. She sent those to Cornell to Rick Hoback, who confirmed they were indeed brown marmorated. The following year, in 2002, they were confirmed in New Jersey, and then we confirmed their presence in Maryland, in Hagerstown in 2003. We found them in West Virginia in 2004, and they continued to spread. Now, there was a bit of a lag period between those introductions and detections and serious injury, and in the eastern panhandle of West Virginia, we actually did not see injury until 2008, when the first reports of late season injury appeared. Then in 2009, we began to see both nymphs and adults in orchards throughout the middle of the season, and there was very serious injury in 2009. And then, of course, in 2010, this was really a watershed year where we saw injury in tree fruit, small fruit, vegetables, row crops, and in vineyards, and throughout the mid-Atlantic states, including West Virginia, Maryland, New Jersey, Delaware, Virginia, and Pennsylvania. Now this is a distribution map of brown marmorated stink bug throughout the 48 continental United States. So the red indicates states where we have seen both severe agricultural problems as well as nuisance problems from the fall immigration of bugs from host plants to potential overwintering sites. The yellow states are highlighted in that they have had reports of nuisance problems in the fall with homeowners, but no agricultural injury reported yet. However, when you see these yellow states, what that is indicative of is establishing populations where the populations are beginning to increase. And then, of course, the green states mean that we've had specimens detected but no injury reported and no nuisance problems reported. And so at this point, brown marmorated stink bug has been detected officially in 33 states and the District of Columbia. Now, the reason we were very interested in brown marmorated, and when I found the first specimen back in 2003, we began tracking it, was because we were seeing increasing injury from our native species, and in the mid-Atlantic we have three: the brown and dusky stink bug, Euschistus cervus and tristigmus, and the green stink bug, Acrosternum hilari. And in tree fruit, this is the typical injury that we find from native stink bugs, where on peaches we see the cat-facing and gummosis early in the season, and then on apple, which was typically late season injury, we see these corky and dented depressions. Again, this is with our native species. And really, the reason for that injury has to do with the changes in our management programs over the years after the advent of FQPA, where we began to shift from our broader spectrum materials to more specific chemistries that were softer on beneficials and that sort of thing. So we were really removing those broad spectrum materials from the system. And what this did was really allowed increased injury from secondary pests like native stink bugs. So during that time, we were developing monitoring tools for native stink bugs to determine the need for and timing of insecticide applications. And here you can see the yellow ground-deployed pyramid trap that we had developed over a number of years. We also were working on attract and kill strategies for native stink bugs as well. And so for a number of years we weren't seeing any injury, as I mentioned, from brown marmorated. However, in late 2008 and 2009, this is what we began to observe in commercial orchards. You see on the upper part of the slide a mature peach with adult brown marmorated stink bugs feeding. And if you peel that skin away, this is the typical injury we would see, this corky, dry tissue beneath. And below is an apple with the large indented depressions, again from brown marmorated stink bug feeding. So in 2009, we were actually having quite a bit of economic injury here in the Eastern Panhandle. This is just one example of a commercial apple orchard, a family-run operation. They have about 1,100 acres. They produce about 500,000 bushels annually, and by 2009, brown marmorated stink bug was their #1 pest, and nearly 10% of all the fruit they harvested, in this case 45,000 bushels of apples, had to be redirected from fresh market to processing due to brown marmorated stink bug injury. And certainly for commercial growers, the loss of value are somewhere between 80 and 90%. So again, already a tremendous pest. In 2009, the other thing that we observed was a tremendously large overwintering population. These photographs were taken on our building here at the lab at the Appalachian Fruit Research Station. So you can see the adults aggregating on the sides of the building. On the lower left, you can see the bumper of someone's vehicle with brown marmorated stink bugs. And this really highlights the fact that they are excellent hitchhikers and can be taken to new areas just because of this behavior we see in the fall, where they aggregate in tight quarters, including vehicles. And then we also had problems with them actually clogging our air intake vent, which you see in the upper right. So what I'm going to do now is sort of take you through what we learned in terms of 2010 and the field season in terms of the different hosts that were attacked and the types of injury that we observed. In the winter of 2010, we had a tremendous snowfall here in the Mid-Atlantic, something we are not very familiar with. But so there was a lot of snow cover, and the question is, did that snow cover in some way help brown marmorated stink bug overwinter more effectively? In other words, we had higher survivorship. The answer is we don't know at this point, but certainly it's a question we have to address. And so What we learned next was by April 2010, much earlier than we had ever observed before, we began to spot brown marmorated stink bugs in some of our experimental orchards here on the farm, but also out in commercial orchards as well. And so we began photographing them just to sort of document that early season activity. But we presumed because many orchard cover sprays are going on at this time, this would protect the fruit from any injury from brown marmorated. So here you can see it's May 5th and they're feeding on Montmorency cherry. Here they are on May 10th feeding on a Loring peach. It's about 20 millimeters in diameter. Granny Smith apple on June 10th. And Seckel pear also on June 10th. And so you're starting to get an idea of the number of different hosts this insect will feed upon. And so again, we were hoping that cover sprays would be controlling this insect, but indeed that was not the case. So by late June, we were starting to see the first evidence of this early season activity in commercial orchards. And in this case, it was very specific to stone fruit. This is a peach that is immature, and what you're seeing on the surface is a bit of gummosis on the left, but when you cut that open, this is something that's very unique to brown marmorated stink bug. We saw these pockets of dead and sort of gummy, dry tissue internally in the fruit because of that early season feeding. And it appears to us as the fruit expanded, these pockets also expanded as well. We also began to observe something that was very different from our native species, which is not only was it adult feeding, but also nymphal feeding. Typically from our native species, we really only worried about the adults. And then the final major difference was that we were learning that, in fact, brown marmorated stink bug could reproduce in commercial orchards. With our native species, they typically would reproduce on broadleaf weeds and then move into orchards, but this was not the case with brown marmorated. So here we are on July 21st, and you can see both adults and nymphs feeding on peaches, immature peaches, in commercial orchards. And you can see the gummosis and injury. Then, by the late July, we began to really see the devastation from this insect. These are some photographs taken from commercial orchards that had total losses of their early season peach varieties. In this case, the grower made the decision to strip those trees and allow the fruit to rot on the ground because it was essentially 100% loss. And you can look up through this orchard, and just every fruit that's on the ground had just extensive brown marmorated stink bug injury from that early season feeding. Then we began to see the other host that this insect would attack. In this case, this is field corn, and so you can see both adults and nymphs feeding through the husk on the kernels, and what happens is the kernels do not develop properly. They also feed on sweet corn, and when they do, the kernels turn brown. Because of brown marmorated stink bug feeding on silk, they were implicated in interfering with pollination. And then we had a number of farmers, as they were cutting the corn for silage, the brown marmorated stink bugs in the fields were so thick at that time, literally they had to turn on their windshield wipers. And so from that, then the question is that silage that was created, would this deter cattle from feeding on it because of the presence of the brown marmorated stink bugs? And if it did not, could it in fact taint the milk because of those defensive compounds from the insect? So this is an open question as well. Now we also saw extensive injury on raspberries where they were feeding on the fruit, and this was both again nymphs and adults, and severe feeding leads to the fruit not coming free of the receptacle, and also we started to see sort of dieback at the tips of the shoots, which again we think may have something to do either with their salivary enzymes or perhaps they're introducing something into the plant that could again be causing this dieback at the tips that we're seeing. They also attack a number of vegetable crops, including tomatoes. You can see the injury on the surface on that ripe tomato, and you can see the numbers. They really do like tomatoes as well as bell peppers. We saw a lot of injury on bell peppers, and you can see the injury, the dry corky tissue beneath the surface. It wasn't limited to vegetables. In this case, this is row crops. And so in the center where you see the part of the field that is referred to as unaffected, where the soybean plants are starting to dry and senesce, this is typically what we would see. But around the edges of fields where you see that green area, that is from brown marmorated stink bug feeding. And the plants do not senesce properly. There's sort of a vegetative response. They remain green. And so the pods do not develop properly. The beans do not develop properly. And this was a major issue for soybean growers here in the Mid-Atlantic. And so the soybeans could not be harvested correctly, and of course there was losses as well. They also, at late August, we see this intense late season feeding period just prior to them entering overwintering. And so on apple, what we began to see was really serious injury. And so this is a pink lady apple, just about 6 to 8 weeks before harvest. You see the fruit is quite deep in the injury is quite deep in the fruit's flesh. And so obviously this is unacceptable for fresh market. And again, if it's redirected to processing, the loss of value can be up to 90%. We have a number of growers that were in this position, and so now the question is, what are the processors going to be doing in 2011? Because this injury is very deep, it's very difficult for them to peel out, and so there may be new requirements. Also, due to the extensive feeding, there may have been questions of premature drop of the fruit. Now we also saw them feeding on ornamental nursery crops. Here they are feeding on Norway maple on the left and a newly planted peach tree on the right. And we don't really know what impact that feeding may have, but certainly with the numbers that were out there, we were concerned. We also saw them feeding on grape late in the season. This became an issue for a number of vineyards who had to get them out of the clusters or the lugs because of issues potentially with taint to the wine. Of course, the feeding themselves causes collapse of the berries, and there were also a presence of increased rots despite the fact it was a very dry year. So those are the issues in grape. We also saw them in nut crops. These are pictures of hazelnuts with feeding injury as well. So you can see the host range is extremely broad. So that took us to harvest. But in fact, the injury did not end there, because throughout the Mid-Atlantic, growers who put fruit into storage at harvest seemingly clean, 4 to 5 weeks later, we're starting to see injury emerge from this late season feeding that occurred during that pre-harvest interval late in the season just prior to harvest. And so it's not visible when the injury first occurs, but several weeks later it appears. And so in the Mid-Atlantic, there's been some surveys done by U.S. Apple with growers reporting, or some of these storage facilities reporting, up to 60% of the fruit coming out of cold storage having injury. So serious, serious problems. So what's been going on? Well, at the national level, one of the issues was just getting the word out. And so back in September, we held a meeting at 2 commercial orchards in Maryland, Catoctin Mountain and Garden Hour. And this really brought together both the leadership from USDA, as well as other government agencies, state departments of ag, and the research and extension community to look at this problem and really get people talking about the issues. And so this was one of the events that really has been useful. Another has been the press, and interestingly, the press really started to pick up on the crop injury at the same time when the homeowner issues began in the fall, when the brown marmorated stink bugs moved to homeowner sites. And probably one of the best issues or best photographs that appeared was in the New York Times. And so you can see the headline of the article, "Move Over Bed Bugs, Stink Bugs Have Landed." And certainly if you're competing with bed bugs for press, you know it's a serious problem. But you can see these folks. This is about a half an hour from our lab. They are literally sweeping up brown marmorated stink bugs into 5-gallon bucket. So tremendous numbers of insects out there. The other group that's been very useful is the brown marmorated stink bug working group that's led by myself and George Hamilton, and this group is designed to bring the research and extension community together. And so what we have been able to do is to develop priorities for research and extension and regulatory. And in looking at this list, you'll probably be able to see that really we are at the beginning in terms of understanding this insect. So this really leads into the research that we've been conducting, and so one of the things we've been working on is developing a monitoring tool for growers that essentially will allow them to detect the presence, abundance, and seasonal activity of brown marmorated stink bug, and then ultimately to design behaviorally based strategies like attract and kill or mass trapping. So let me quickly take you through some of the work that we've done toward a monitoring tool, looking for visual cues that can be incorporated into a trap design, olfactory cues, the lures we use, a capture mechanism that is compatible with their uh, movement patterns, and then a deployment strategy. Where and when do we deploy these traps? And so what we have done so far is look at visual cues associated with a pyramid trap, one that we had used for our native stink bugs, and we evaluated these 5 different visual stimuli sources with baited and unbaited traps over a 2-year period. And so we were able to capture a lot of stink bugs, obviously, and what we found was that for both nymphs and adults, a darker pyramid that has baited with a known attractant is by far superior. So here you see adult captures in 2009, a baited black pyramid trap. In 2010, there was no difference between our dark green and black, but with the nymphs as well, we're seeing a baited black pyramid trap being superior. So that's the first piece. The second is what we're doing right now is the fact that brown marmorated stink bug interestingly respond to wavelengths and intensities of light. And so in this case, what we've been doing is trying to discern what are those wavelengths and intensities so we can incorporate it into a trap design. This picture, by the way, was taken at a bank in Spring Mills, West Virginia, and the size of the aggregation around this light was estimated to be about 1 million individuals. So we have a light bioassay right now that's trying to capture that information, and so you can see where we're releasing insects into this arena and tracking their movement and determining those wavelengths and intensities. We also are trying to identify the true pheromone. These are headspace collection chambers that allow us to identify the headspace volatiles being released by, in this case, adult male bugs. And so we hope in collaboration with Dr. Aijin Zhang, who's leading this part of the project, we'll have a better attractant that we can use with our traps to really lure these insects in. So that's another piece of this puzzle. In terms of capture mechanisms and deployment strategies, interestingly, there are a few commercially available traps from Asia that we were able to get our hands on and compare with our prototype black pyramid trap, which you see on the right. So in this case, the traps that were available from Japan, they were different in that on the left you see this clear sort of pyramid trap you deploy in the canopy of trees compared with the ground. The center is this yellow water trap that's considered to be visually stimulating to the insect and again deployed in the tree. So we were able to compare these at our farm, and what we saw was in this case that our black pyramid trap was superior in terms of capturing both adults and nymphs, and so compared with these commercially available traps. So again, this is where we are right now in terms of being able to monitor adult and nymphal populations. So just to wrap up this portion, we are using this dark visual stimulus as our part of our trap design. It's a ground deployed pyramid because both adults and nymphs have a tendency to walk up surfaces. That's their natural behavior. We have a known attractant. It is particularly good for the nymphs season long, but the adults really don't respond strongly until later in the season. So identification of a pheromone is really critical. And then we hope to augment these trap designs with light based on optimal wavelengths and intensities. Now, the last piece I want to talk about with you today before I wrap up is just the challenges we've observed in managing brown marmorated stink bug in commercial plantings, because one of the issues is that brown marmorated stink bug has an incredibly broad host range. They feed on over 300 different plants, and what this means is tremendous season-long pressure from populations moving from wild hosts into cultivated crops like our commercial orchards. In terms of managing them, we've seen greatest efficacy when adults have direct contact with finished wet spray material. However, because they're constantly immigrating into plots, only a small population have contact with that material, and so that's a problem. Also, they have incredible avoidance behaviors that allow them to potentially escape treatments. And finally, insecticides that are labeled as excellent against native species have not shown the same efficacy against brown marmorated. In fact, we have seen knockdown and recovery from a number of compounds and other compounds being completely ineffective. So let me show you this knockdown and recovery phenomenon. So what we did was to treat 1-acre plots with tombstone, which is a pyrethroid, or vidate, a carbamate. We recovered these bugs from these plots and followed their mortality over time. And the reason we did this is because in grower orchards where we saw them spraying pyrethroids, particularly like Warrior and Asana, literally 80% of the bugs recovered within 24 to 48 hours and were climbing back up into the trees. As incredible as that sounds, that's what happened. So in this case, from this small bioassay that we did, what you can see is we tracked mortality over 5 days. And so at the time of collection, all of the bugs were moribund, meaning they were unable to move, but they were still alive. Their antenna or leg might be twitching, but they could not climb vertical surfaces or move on horizontal surfaces. So red is dead, and alive means they are capable of moving once again. And so you can see that with tombstone, by the end of the trial, about a third of them had recovered from that exposure. And so this was the issue. So what we did was to think about that in designing an insecticide evaluation that we conducted over the winter months, where we really needed to deal with what we considered to be the primary threat, those insects that come in after the spray residue is dry. So in this case, what we believe is control will depend on residual effectiveness and likelihood of uptake. So our laboratory bioassay was designed to be biologically relevant based on that primary threat. And so all of the compounds that we evaluated through this 3-part trial required about 24 person hours. So this is the list of materials we have evaluated. We evaluated both carbamates, organophosphates, pyrethroids, neonicotinoids. We looked at the organochlorine material thiodan or endosulfan, as well as other classes and newer chemistries. And this 3-part bioassay included, first of all, using EthoVision, which is a software package that allows us to track their movements on insecticide-treated dishes in a horizontal plan. So it tracks horizontal mobility. And we would conduct these trials over a 4.5-hour period where they were exposed to these dry residues that had been cured for 18 hours. Then we would remove them from these dishes and then look at their capacity for vertical movement to sort of mimic what may happen if they were climbing back up into the trees. And then we tracked mortality for 7 days. So I can't go into all of these results, but what I want to show you is the type of tracks that emerged from, first of all, the horizontal mobility, where you can see on the top with water as a control, the bugs kind of meandered around the dishes. There wasn't really any impact. But with Warrior, a pyrethroid, you can see at 0 hours they appear to be very stimulated. Within 1 hour, you can see the track becomes more irregular, they're succumbing. And within 2 hours, you can see it looks like the bug's just about dead. And that's what we would have interpreted. But in fact, because of that knockdown and recovery, all of those bugs, 100% of those tested recovered. And in fact, they could even reproduce. So we tracked bug mortality for 7 days, and this provided us with this lethality index. And I know this is a long list of materials, but what I wanted to point out is that we ranked each of these materials based on how quickly they succumb to the material and also the overall rate of mortality. And so you can see that a lot of the materials are older, broad-spectrum materials that were most effective. So to wrap this part up, in terms of the insecticide work, broad-spectrum materials including dimethoate, malathion, bifenthrin, methodathion, endosulfan, and methanol provided the greatest levels of contact lethality. Now, certainly within each class, the patterns of lethality are similar, but there's a substantial amount of variation between active ingredients in terms of the intensity of effects on mobility and mortality. Based on the activity of registered and unregistered compounds, Section 18 for dinotefuran, also known as safari or venom, is being pursued for tree fruit, with Virginia as the lead state. This is a mid-Atlantic regional application. And in terms of tree fruit and other crops, recommendations are being put together based on what we have in the Mid-Atlantic, which is a season-long threat. Now, I know in other areas it'll probably still be more of a late-season threat, and in other areas, obviously, it isn't well established. So in terms of where we are right now, we have formed a nationwide team of 50 research and extension personnel throughout the U.S. representing 14 institutions in 10 states. We've put in a very large specialty crop research initiative proposal entitled Biology, Ecology, and Management of Brown Marmorated Stinkbug and Orchard Crops, Small Fruit, Grapes, Vegetables, and Ornamentals. And in terms of my lab's work, we're going to continue to work on the monitoring tools and behaviorally based management strategies. But the bottom line for brown marmorated is that it's going to require a sustained, cooperative, collaborative, and integrated approach for research and extension on a national scale. And I will tell you that certainly within the strawberry industry, folks are going to be looking for brown marmorated in 2011 and trying to document any problems. So far, none have been reported, but with the broad host range, we have to be extremely cautious and certainly document the host that this insect may attack. So this is my lab group that work with me on all of the projects that we're conducting, and they're a great bunch of folks. And then finally my colleagues and growers and others throughout the Mid-Atlantic and the U.S. who have worked with me on this project. So I would be happy to take any questions, Kathy, if there are any.
Thanks a lot, Tracy. Are there any questions? Okay, we'll give you a chance to think about that, and Tracy will be back with us after Stephen does his presentation. Our next speaker is Mr. Stephen Koike. Stephen joined the Monterey County office of the University of California Cooperative Extension in 1989. As a plant pathology farm advisor, Steve is involved in educational programs and applied research in vegetable, fruit, and ornamental crop diseases. His research emphasizes diseases of vegetables, epidemiology of disease systems, fungicide evaluations, identification of new diseases, and ecology of fungi, exploration of alternative disease control methods, such as alternative fungicides, biological agents, organic agricultural disease management. Steve is also involved in applied research of foodborne pathogens, primarily E. coli, as they occur in agricultural field settings. Educational efforts consist of informing clientele of research results, tailoring results to commercial operations, and diagnosing problems found in field and greenhouse. Steve operates the University of California Cooperative Extension Plant Pathology Diagnostic Lab located in Salinas, and in 2007 he published his first book, A Color Handbook of Diseases in Vegetables. Steve?
Great, thank you, Kathy. And I see there's some questions about the stink bug. Did you want to take those, or should I just go ahead?
Oh, okay, I didn't see those. Let me see.
I'd be glad to wait if you want to do those.
Yeah, we could do that for just a second. Okay, Laura McDermott would like to know, Tracy, if brown marmorated stink bug is a pest of blueberries.
Yes, so Dean Polk at Rutgers did document them feeding on the late season variety Elliott in 2010. Other than that, there hasn't been, I mean, they're going to be doing season-long monitoring this year, so that's what I can tell you at this point.
Okay, Pam Fisher would like to know how it moves to new areas.
Generally, we think that, first of all, they are spread in some cases by their fall overwintering behaviors, in that they end up in people's belongings and they move them to a new location. But certainly we've also seen, as they established here in the Mid-Atlantic, that they move themselves. You know, that leading edge of the population has continued to move through our region and head south. So both of those are sort of providing the spread, I would say.
Deb Moreau would like to know if your pyramid traps or patterns thereof are available for other collaborators.
They are. We have a blueprint that's available, and also there is a commercial provider that is making them this year, Ag Bio, which I can provide that to you, Kathy, if you want to pass both the blueprint and that information on.
Yes, that would be great.
Okay.
Bob Martin is wondering where they overwinter, and can they be targeted at the overwintering site?
Yeah, that's an excellent question. We know that at least some portion of the population certainly ends up in structures and homes and things like that. But in nature, it's a big question. What are the preferred overwintering sites? We've done scouting and found them under sort of bark on trees, as well as sort of in rocky crevices and things like that. And that's where they've been reported in Asia as overwintering. But again, we don't know. And so we have some experiments designed this year to sort of set up both understanding where they overwinter most effectively and in terms of survivorship, but also trying to track their movements to potential overwintering sites.
Carla is wondering if there's any influence of temperature or weather on insect movement.
Yes, both at the high and the low end. We don't know exactly what those thresholds are, but at lower temperatures, we see them more than likely just walking around, so it's definitely, I don't know what that threshold is. And even at higher temperatures on some of our hotter days, these bugs would hunker down and sort of just rest. But again, we have to define what those are.
Okay, and the last one for now, Bob is wondering if they have parasitoids for these insects in Asia.
Yes, they do. There is a species of wasp in the family Selenionidae, genus Trisulcus, that has been reported to parasitize up to 80% of the eggs. And in fact, those have been collected and are in quarantine at our sister facility in Newark, Delaware. Dr. Kim Holmer has been screening these potentially for release within the U.S., but again, we're still very early in that process.
Pam Fisher commented Bob would probably like to go on a collecting trip. Okay. There'll be time to ask more questions of Tracy at the end. Steve?
Okay, thank you. Well, first I want to thank Kathy and the organizers of the webinar for inviting me to participate. And I appreciate the chance to share some things that are happening out here west in California. But first of all, I want to say I really enjoyed Tracy's presentation, learned a lot, and it makes me glad I'm a plant pathologist because some of those pictures were creeping me out. I don't think I would like to see all those bugs around my house or my vents either, so very interesting, Tracy. And I think my talk is very consistent and very appropriate to put with Tracy because I'll be talking about some recent challenges to strawberry out west here, which also involved new problems and perhaps recent introductions. So some of the principles and things might really mesh well with what Tracy talked about. So first, I think the audience here is probably very experienced, but just to pull this all together, I'm going to be talking about some soil-borne issues, some new soil-borne issues on strawberry. And so here's a picture just showing us to remind us that soil-borne problems, by definition, are plant pathogens that are in the soil. So at the top left, you can see what's called lettuce dieback disease on lettuce. It's actually a very strange pathogen. This is a soil-borne virus. The virus is in the soil. It's spread in the soil and the water, and it is very aggressive and causes significant problems on certain types of lettuce, like this romaine picture here. And the bottom left, here's a soil-borne problem on onion. This is white rot, a very destructive disease on that group of plants. And then to the right, verticillium wilt on strawberry, probably a very familiar disease to many of you, and again a soil-borne problem. So we're just going to be talking about these new issues that are soil-borne issues in California and on strawberry. And just to pull together the basics of this type of issue, again, I think a lot of you in the audience already know this, but there are some very distinctive features of these challenges that are in the soil. First of all, they often survive for a very long time. These pathogens in the soil sometimes can actually survive for decades without a host plant there. And as you might guess, that is a tremendous headache for a producer, for a grower or farmer, because even if you're not growing the crop, the fungus or the pathogen is still there. So they're very long-term survivors in the soil. And they're often difficult to detect. That's not always the case. We have some good tools for some of these, but in terms of trying to know whether your field has one or more of these problems, it's not always that easy to find out before you plant the crop and before you see the problem occur. So it's sometimes difficult to detect and find them. And I probably would add to the slide, they're also difficult to diagnose. I'll touch on that later in the presentation, but sometimes knowing which one you have is often not easy. As I mentioned, these would be very damaging. You're talking about plant death, or on the brighter side, just crop loss in terms of yields. These problems, again, are very significant economic factors in agriculture. And part of the reason why they're so important and damaging is that they're easily spread. Not quite like stink bugs, but we spread these ourselves, meaning we as farmers and growers and people. For the most part, these are primarily spread as contaminants in dirt or mud and on equipment, on tractors, on trucks. So probably the growers and the pest control advisors and field walkers and all these people who are moving equipment and vehicles around, that's how we spread a lot of these. We can also spread them in propagation material, that's another factor, but most of the time we're spraying them in the dirt in which they're found. One problem in terms of managing soilborne problems or pathogens, for the most part, fungicides are not very effective. We could probably find exceptions if we look around, but for the most part, your Fusariums, your Verticilliums, all these many soilborne problems don't respond well and are not really controlled well by fungicides. And then often these pathogens have a wide host range. So you may be growing strawberry or crop X. You'll say, well, I have too much of a problem, this soilborne problem on strawberry, I'll plant something else. Often that other crop can also be a host and will prolong that problem. So it's a complicated set of factors here, but this one slide kind of summarizes these features of soilborne pathogens. So the issue that's happening out in California is a fairly recent one, and I'd like to walk you through what's happening out here west regarding this. But I would say that even if you're not out west here and you are a strawberry grower somewhere else in the country, or you grow some other crop and you don't have these issues, I still would argue this might be of interest to you because it kind of possibly mirrors or acts as a model for what happens when new pathogens are introduced to an area and when new problems occur and how you approach it, how you diagnose these things. So I think, again, even if you're not sharing our issue particularly at the moment with Californians, the information and the history that I'll show here certainly can be reproduced in your area with a different crop or a different pathogen. And by recent, what I mean is the dieback and collapse issues on strawberry that are new, we first began to see in 2005. And unfortunately, from 2005 through the present, we've seen an increasing incidence, meaning we've seen these problems spread throughout the state of California. If in one county we had 2 fields that had the problem in 2005, by 2010, there were 12 fields, so we're seeing more and more growers and more ranches affected by that. Very important to point out, these new dieback and collapse problems, which I'll show you, are not associated with and have never been associated with the other well-known pathogens that already occur in strawberry. So if you're a strawberry person, you know that there's Phytophthora issues and there's Verticillium and there's Rhizoctonia. You know there are some well-documented historical soilborne pathogens on strawberries. These new problems I'm showing you today have never been associated with these other problems. And the other important association here, almost without exception, these are occurring in fields that no longer receive the traditional fumigation treatments. So in California, of course, you know, our industry has really relied on methyl bromide and chlorpicrin flat fumigation, meaning we're applying that fumigation to the entire field, not just to the beds, and that practice has really made it successful in terms of yields and dollar value. These new problems are occurring in these fields that no longer receive that traditional flat fumigation, either because of cost or, of course, the change in the regulations and with methyl bromide leaving on its way out. So those factors not associated with previous pathogens that are known to occur and occurring where we no longer fumigate in the same traditional way, these are really hallmarks of these new problems. Now today we know that there are 2 major pathogens associated with these problems, Macrophomina, Phazulina, which causes a disease called charcoal rot, and a strain of Fusarium oxysporum, which causes Fusarium wilt on strawberry. Neither of these were known to occur on strawberry in California prior to 2005. In terms of the history, again, what's happening, initially I'll show you some maps, but initially we only saw these problems in Southern California where we have our southern crop, and now it's occurring in other parts of California, and I'll illustrate that for you. And then in a second here I'm going to show you what it looks like if you're not familiar with these diseases, but the symptoms are very much characterized here by this list. You see the wilting of plants, there's overall poor growth and performance, and then as the disease progresses and as you go into the season, eventually the plants will actually collapse and die. And then when you examine these plants, it's very important to dig them up out of the ground and cut them open and look inside the plant. You'll see a very distinctive discoloration of the inner tissues, primarily the crown. So let's take a look at these problems. This is the first one. This is charcoal rot caused by the fungus Macrophomina, and this is a soil-borne fungus that's known to occur in soil. And I believe you would share my opinion that this is a significant problem. If you see this type of photograph, or if this is your field, we have distinct areas where a large percentage of the plants collapse and die, as pictured here. Just a little closer up look to show you again Macrophomina or charcoal rot occurring on strawberry, and plants, especially the one in the bottom center photo, showing advanced cases where they're collapsing. The photo in the upper right, and let me try, Kathy's trained me on this arrow here. Hopefully you can see that. The photo on the upper right, this is a collapsed plant, and I point it out to you now because later I'll talk about diagnosing and trying to identify these problems. And here I would argue and debate with you the fact that this type of symptom looks very much like it could be infected with verticillium. So again, if you're looking to say identify this just based on the picture or on a visual of the plant, I think that's a dangerous assumption because this is Macrophomina and not verticillium. As I mentioned to you, to diagnose this, or to assist in diagnosing it, you need to dig up these plants and cut them open. And the Macrophomina always shows a very distinctive discoloration in the crown here. You know already that the healthy plant tissue will be cream colored or light colored like this, but the infected plant with Macrophomina will have these very distinctly dark discolored sections of the crown. And here are 3 examples of that. Now what we do here in California, of course in all states where you have plant pathologists like myself, you give these plants to a plant pathologist and they'll take out, through a sterile procedure, they'll take out small bits of that crown tissue and put it in a petri plate with appropriate media, and that's what you see in the upper left here. And that black speckly colony growing out from those strawberry pieces, that is the Macrophomina, and this is the way you confirm the presence of this pathogen in strawberry crowns. And then I mentioned earlier, one reason why these pathogens like Macrophomina are so dangerous and bothersome, they last in the soil for a long time. And here in the large photo in the front, you can see why. Every one of these little black specks is one survival structure. This is called a microsclerosum. And every single one of these little black specks is it's like a seed for the fungus. It is very hard, it's black, which means it's able to resist ultraviolet light, and it's able to survive and persist in the soil for a prolonged period of time. And this is what the fungus makes to enable it to survive to the next crop, to the next strawberry crop. This particular photograph shows the size too. There's my trusty penny at the bottom, and this is grown in a sand and cornmeal medium. So here you can see these little crystalline pieces here. These are little grains of sand. So this gives you an idea of the size of the microsclerosum, how they're very small, and how, if the fungus is making these are easily transported in contaminated soil. These can be in the soil in just a clod of mud on a tractor or a disc, and when you move that disc to a clean field, you've moved these microsclerotia. So this shows again why that's a problem for our growers and people in ag. Okay, so that's the first problem. The second one is Fusarium wilt, and it's basically following the same pattern here. Recently introduced to California, not seen before in California, soil-borne problem, spread in the soil, and we began by seeing it in small patches. The photograph at the top shows what we began to see in, I think, 2006 actually, where these small areas in our strawberry fields began to collapse and die, and over time these patches got larger. The photograph in the bottom on the front shows a couple years later where these patches have gotten much larger, more plants are affected, and we're beginning to be very concerned about spread to new areas. Here's a close-up of that. This is Fusarium wilt on strawberry. Again, it's our second new development in fields that are no longer receiving the traditional flat fume treatment. And in a second I'll talk about diagnosing these, but again I would argue that you and we cannot separate this photograph from the macrophomina ones. The plant symptoms are identical and also resemble verticillium wilt. Again, you need to dig up these plants and look at them and cut them open. And again, just like the macrophomina, the Fusarium wilt causes a very distinctive dark discoloration in the inner crown tissue. And so again, one cannot diagnose this strictly by looking at the outer collapse. You have to dig up the plant, cut open the crown, and look for this type of symptom inside. Now, as I've just shown you with macrophomina, I'm telling you this is Fusarium. So you say, well, how can you separate these? Well, you cannot separate, again, based on the symptoms. So you take these plants to your friendly neighborhood plant pathologist, and we will, again, plate pieces of that strawberry tissue on the appropriate medium and look for what grows out. And in this case, this is the Fusarium, which has a different look to it. It has a very pinkish color in this medium, and we can diagnose it based on this recovery. The photograph in the lower left shows you the survival structures for this fungus. These are called chlamydospores. It functions very much the same as those black specks of the macrophomina. It survives in the soil for a prolonged period of time. It resists weathering, and this is how the fungus survives between crops and from year to year. So to show you our dilemma, or our issue and concern, here are two snapshots in time, as you will, for our state regarding macrophomina. In 2005, we began to see, detect, and confirm the macrophomina on strawberry in California, and it was only in 2 counties. We have a significant strawberry industry in Ventura County and Orange County, so those are the two pink stars there. So 2005, we began to get concerned for our Southern California growers because of this problem. Unfortunately, when we fast forward now to 2010, we have a number of other counties that also report and have confirmed this same macrophomina on strawberry. As you probably know, our major area of strawberry production in California is the central part of the state all the way down through the south. So this is Monterey County, actually where I'm located here in Salinas. This is Monterey County. From Monterey County south, we have the majority of the state's strawberries, and you can see that 4 out of the major 5 counties for strawberry in this area have the macrophomina. The 4 counties to the north have smaller, more limited strawberry industries, so the acreage is limited, but they also have the problem. So you could see why we're concerned about the future when you have this spread and increasing incidence throughout our state. So that's macrophomina. And then here I want to show you a similar pattern, although at this point fewer stars, thankfully, for Fusarium wilt on strawberry. 2006 was the first year we confirmed Fusarium on strawberry in California, and you could see that the only place we found it was in Ventura. And then we fast forward to 2010. We have 2 sites, 2 counties with the Fusarium. That's good news, bad news. The good news is it has not spread to, say, 10 or 8 counties. The bad news is, however, the Fusarium has been found in one of our major strawberry producing areas, and that's my home base in Monterey County here in the central coast of California. So even though there are only 2 counties affected, these 2 counties probably account for, I would estimate, 65 to 70% of our state's strawberry production. So even though it's not statewide yet, the Fusarium has touched down in our 2 major producing counties. In terms of the distribution as of last year, just for background information, macrophyllomania is not a new disease in California only. It's also occurring in other parts of the world, and I might add, it's very interesting to note that for all these other parts of the world reporting macrophyllomania, it's primarily showing up again in locations where they no longer use methylbromide. So Egypt, France, India, Israel, and Spain, these places are reporting macrophyllomania on strawberry, and these are places where they are not using the traditional fumigation practice anymore. For macrophyllomania, we need to add Australia. Australia is another country that has macrophyllomania on its strawberry. In the USA, maybe some of you know about this, I don't know where you all are from, but right now in strawberry, it's been confirmed and reported in California, Florida, and Illinois. And then I talked to you earlier about the counties in my own state with that map. For the distribution of Fusarium, Fusarium is slightly different than macrophyllomania. Macrophyllomania is a new worldwide development, and what I mean by new is, in the year 2000 and counting, that's when macrophyllomania began to be reported on strawberry. Fusarium is a little bit different. Fusarium is not a brand new disease because I believe it's the Japanese and maybe the Koreans reported Fusarium back in the 1960s and early 1970s, and I think Australia also has had it a long time. So Fusarium mold on strawberry has been in the world a longer time, and unfortunately now we in the USA are just added to that list of infested countries. But for us it's brand new in California. For whatever reason, the Fusarium wilt disease on strawberry has been reported in California and South Carolina in terms of USA states. So let's talk a minute here about something that I think is extremely practical for almost all of you probably participating and listening in today, and that is diagnosing. If you're a grower or a pest control advisor, or you walk fields and make recommendations, or you're a salesperson selling products to farmers and people in ag, you know that it's extremely important and critical actually to identify and know what the problems are. I mean, if you see a stink bug on your crop, you better call Tracy to find out which one it is, right? You need to know what the problem is and have a precise identification of the problem. And for pathogens that originate in the soil, that's very much a challenge. And as this photograph here illustrates, I'd be glad to bet with you your lunch money about which pathogen or pathogens might be responsible for these symptoms. The point is you can't tell. And I, as a professional who work with these 12 months of the year, I cannot tell which pathogens are responsible for these dieback problems, because it could be one of our new issues, macrophyllomania or Fusarium, or it could be one of the old-time favorites, Verticillium phytophthora. We do not know. And I want to use just a couple minutes here to illustrate why this is a very big challenge and why it's important to recognize the issue. So the first challenge is with our new issues here, our new problems. The macrophyllomania and Fusarium, in my mind, are virtually identical in terms of the symptoms. Poor growth, stunting, dieback, plant collapse, discoloration of that crown, which I showed you, and also haven't talked about this too much, but very much stress related. Both of these pathogens can really cause the most severe damage when the crop is stressed, and that is either through environmental extremes, when it's very hot and dry, or there's a little bit of water stress. If you're a grower and you're half a day or a day late watering and the plant's a little bit under stress because of that water shortage, these pathogens are very aggressive and cause much more damage and much more rapid development of problems under stress. So for macrophyllomania and Fusarium, both of these act the same, they look the same. You and I cannot separate them readily without a laboratory test. So diagnosing these are going to be a problem for you, they're a problem for me. And then to expand this a little bit, I mentioned the old-time favorites, Verticillium phytophthora. For the most part, these symptoms and features are shared by all four of these problems. The two new ones, macrophyllomania and Fusarium, and the old ones, Verticillium phytophthora. So if you have these types of symptoms, in general, it's going to be very difficult for you to know what's going on without the support and help of a laboratory or an extension pathologist or someone who can actually have things tested. The one exception here is Phytophthora is really not considered a stress organism. You can have phytophthora issues on strawberries that are growing quite well. And in fact, as you know, being a water mold, it likes lots of moisture in the soil. So the stress thing is not quite relevant here. There's some good questions here. I think it'll be a good chance to take these right now. Are these problems worse under plasticulture? Maybe Teresa can send me a follow-up question. I'm wondering if you mean plasticulture, meaning large hoop houses that cover the whole planted area, or plastic-covered beds that the plastic is just covering the beds and the plants are planted into that. So I need that. Have these diseases ever been observed in fumigated fields? We have seen them in fumigated fields. Again, we need to qualify this here. In California, our experience is primarily with the production fields. These production fields have been fumigated with alternatives, not the methylbromide chlorpicrin. They have been fumigated with these new replacement materials. We are seeing these diseases in those situations. If it's fumigated with methylbromide chlorpicrin at the appropriate rate, and that's the traditional treatment, we do not see and have not seen those problems. So yes, we are seeing them in fumigated fields, and these are ones that, again, are in the new world being treated with alternative materials. Okay. And then finally, a question about nursery stock. As of right now, and I think we need more information from the nursery producers in our state, we have not heard of significant problems of these two new pathogens in the nursery production counties. Have not seen that. And we do receive materials from them for testing for other things, and so far I've never isolated Macrophomina or Fusarium from transplants coming from those areas. We do get the old standards, the Verticillium Phytophthora, Colototricum, but no, we have not seen these new ones from transplants yet. Okay, Teresa got back to me. Let's see, the question is, okay, so I think she's asking, do we see these problems, or are they worse in fields where the beds are covered with plastic and then the plants are planted into the plastic, as opposed to the use of straw or mulch as a covering of the bed? For us in California, you may know this, we do not use anything except plastic. We do not use straw or mulch covers for strawberry production. So we only see it in the beds with the plastic, but that's because all strawberries are planted into plastic covered beds. So as of right now, I don't know how that dynamic might play out for other types of bed covering. And then the last question, then I'll move on for a second here. The question is, has Macrophomina, let's see, question is, can Macrophomina be a problem in a temperate climate zone? Again, I'm only a mere plant pathologist, so you need to define for me exactly what temperate climate means. Does temperate mean me here in Central California, Mediterranean type climate? I'll look for the reply on that. I'll go ahead here and get to that question in a second. Okay, so we in California have these problems. Hopefully you won't, but the way things move in the world and with these new developments, perhaps you will. So what are we collectively going to do about dealing with these problems? Again, I think an audience like this today, you're probably very experienced, so you could do this slide, you could put this together. You would approach the Macrophomina or Fusarium issues the same as you would any other soilborne issue. That's nothing novel about that. What is novel is the fact that we have new pathogens on strawberry in California, and what's novel is we're in an era where we're losing the main tool, methylbromide, that has been used in years past to defeat these challenges. So that's what's novel. What is not novel is how you deal with it. It's going to be the same as with other soilborne issues. So first of all, select the appropriate site. I mean, if you have the flexibility and the ability to just move off site, don't plant in an area that reports a history of this problem. If you know that this field over here down the road has Macrophomina, then you would not be well advised to lease that ground and plant strawberry next year. Stay away from known infested sites. Very important is crop rotation. If you're a diversified grower and you grow things more than strawberry, just don't plant something that's a host. Don't plant strawberry. Don't have time to go into this today, but we know or feel that this fungus will affect other crops that are known to be hosts of macrophamina. So examples, pepper. So if you have diseased strawberry in a field, don't plant pepper there either. And don't plant strawberry anymore. Try and rotate crops. Plant lettuce, plant broccoli, plant petunias, plant things that are not a host. Rotate away from that problem. Rotate away from strawberry. For conventional growers, fumigation remains a tool, a management tool, but the question is which materials. I'll show you a slide near the end here that shows our work in California that indicates some of the alternatives do control the macrophamina and Fusarium, but it's not 100%, and we are getting escapes, and we're getting a reinfestation of these problems, so that's not the answer in itself. We do have fumigation that will help, but it will not eradicate the problem because it's not as good as methylbromide chlorpicrin. If you have access to resistant strawberry cultivars, that is obviously a good management tool for soilborne problems. I'll again show you at the end some of our work. We show that there are differences in the varieties, and so we have the ability to maybe in the future breed for some resistant varieties. As of right now, we don't have true resistance, but we have varieties that are more tolerant. Plant those if those are available, if they fit your situation. As I mentioned to you, both the new problems are definitely stress-related. Plants that are stressed from environmental extremes, and I think I forgot to mention fruit load. Plants that are stressed with a very, very heavy fruit load, that's good for you, the grower. That's bad for a strawberry plant that's already infected with macrophamina. A heavy fruit load puts a physiological stress on plants, and that will definitely create the onset of very severe and fast-developing symptoms for these problems. And finally, of course, we talked about soil. If you are moving dirty soil, infested soil, from place to place between fields, you are going to move these pathogens, so stop that. Don't do that. We're having more and more growers actually wash off equipment. It's a big pain in the neck, but it's something that is extremely important if you want to safeguard clean fields in the long run. Okay, let me quickly take a couple more questions here. Okay, great question. There's a question about whether you can have this problem show up in cooler climates like Northern Europe, Poland, Germany. Very good question, and I would say, of course, we never want to say never, so we can't say that, but we can say that macrophamina and Fusarium are both well known as warmer temperature pathogens. So both of these pathogens prefer warmer soils, and I think, I won't go back here, but you can see from the slides, in general, a lot of those countries that report the problem tend to have areas where the climate's very warm. So as of right now, we haven't had any of these reports from, say, Norway. So I would think that the cooler climates would either not have an issue with these on strawberry, or if you eventually have the issue, the severity of the problem may be lessened because of the cooler climate. Both these pathogens like warmer temperatures, and I know that's relative, but still I think Northern European production might not have the same issue. Okay, and I can't remember if Tracy, I think we got Tracy's question. She's going to be leaving us soon. Question from Bob. Does this problem occur in soybean Illinois or in soybean anywhere, I guess, where plants are under stress? And yes, the answer is yes. Macrophamina is a well-known pathogen of soybean. So soybean has been a known host of macrophamina for a decade or longer, probably a long time, and so that's very much a known part of the problems for soybean. And I don't know about the stress thing for soybean, but I imagine the profile is similar. If the soybeans are under water stress or other issues, we do tend to see macrophamina develop faster on any crop under stress, so definitely yes to that. Good question. So in the time I have left with you, just a little bit about our research to highlight what we discussed earlier. So there are differences in strawberry varieties in terms of susceptibility to the macrophamina, and we're doing similar work with this on Fusarium as well. Just want to show you a little bit of information here. So for example, in this scale, a bigger number is bad. So Diamante here at the bottom, the maximum is 5. Diamante has the 4.9 disease severity ranking. That means that it's extremely decimated by the macrofomina when you infect it. However, the Seascape has a 2.9. It is significantly more tolerant to the Fusarium, to the macrofomina. It holds up much better, but I hasten to add that it has a 2.9. We would like to see a 1.0 there. A 1.0 means basically no symptoms, the plant is not affected by the macrofomina, and we are looking at a resistant plant. Seascape is not resistant, but it is more tolerant, so it holds up better, but it is still diseased. But this shows you that our hope is to develop varieties that are resistant, and so that we would have a chance to develop that and replace the ones that are very sensitive. Bob is asking, are these numbers, the severity index numbers here, related to crop load? In this particular experiment, no. In fact, I'll show you the next photograph. This is a controlled experiment in a shade house. We inoculated potted strawberry plants just to see the basic reaction to the fungus, and this does not have anything to do with harvest fruit load or just the fruit load of the plant itself. So no, it is strictly susceptibility of the plant to the fungus. And this is what I'm talking about, Bob. So here is our Seascape on the far right. It has been injected with the macrofomina just like the other varieties, but you can see that even though they're inoculated at the same time, the Albion and Ventana are much more sensitive to the macrofomina. The Seascape holds up much better. However, if I gave you the Seascape plant and you cut it open, you can see slight discoloration in the crown, meaning that it has been infected, and if you wait long enough, the Seascape will start to show collapse problems as well, but it will take a much longer time to develop. And this is kind of summarizing the work overall in terms of some of the resistant varieties. So let's look at the column here for macrofomina. I just showed you Seascape. Seascape and Chandler are two strawberry varieties that we're calling it resistant. Resistant does not mean immune. Immune means you don't even get infection, but they are infected. But Chandler and Seascape hold up much better to macrofomina than, say, the varieties Monterey, San Andreas, and Ventana, which are extremely susceptible or sensitive. In the other column, under Fusarium, however, the Chandler and Seascape, unfortunately, are very susceptible. So if you have both of these problems in your field, one or the other is going to get you, unfortunately. If you inject these plants with Fusarium, the Monterey, San Andreas, and Ventana are resistant. So that's good news, but of course, as I mentioned, they are susceptible to the macrofomina. So we have the unfortunate situation where there's not yet one variety that will withstand both of the new challenges in California. Okay. Then finally, just to talk about chemicals. So the deal is, as I mentioned, we're losing the methylbromide chloropicrin. The standard methylbromide chloropicrin at a high rate used as a flat fume application in California to treat the whole field, that is going out. And we strongly believe that is why these new problems are coming in. The old traditional means of controlling is going out, so the new pathogens are coming in. So we have looked at alternative treatments, the list is here, to see if there's something else that can really help us in combating these problems. And two things to note here. One, there's a lot of different treatments. I won't take the time to go through this, but a lot of different treatments. This is one of three experiments we're doing in this type of research to find an answer to these soilborne problems. So we have a lot of alternatives we're looking at. Secondly, the photograph is important to remind me to tell you, these are applied to the bed. In California, we're getting away from applying it to the whole field. The public opinion, the regulators, the general direction of the world is away from using all these chemicals at high rates and high volumes. So one way to address that is just to apply it to the beds. The beds are preformed. They're almost ready to plant. The fumigation material, the fumigant material, is then injected or applied to the beds, sometimes applied through the drip irrigation that's in the bed already, and then later, after time has elapsed, the plants are put into the beds. That's bed fumigation, so we're applying these materials in this system, in this bed fumigation method. And again, time won't allow me to do this or go through this individually, nor do you want me to do that. The bottom line is, if we saw something that was equivalent to a methylbromide chlorpicrin traditional fumigation, the bar would reach the number 1 here, but go no further. We are looking at trying to get something equivalent to a level 1 in terms of disease severity. You can see the good news here. The good news is a lot of these alternatives, the first 9 or so, control the problem quite well, and so they're doing a good job at controlling or at least knocking down populations. The bad news is they are not comparable to that level 1. They all allow some macrophomina and some fusarium to creep through, and that's what we're continuing to see in these fields. We do see dieback and collapse, even though the plots have been applied with these materials. And you can see controls number 10, over here way on the right, that's something that is not acceptable, and we have 3 treatments that are about the same as a control. So our situation in California, we say none of the new alternatives, some of which can already be used, will give you the same level of control, and we are seeing breakthrough in some disease occurring despite the use of those new alternative materials. So I think I'm about done here. In summary, I've walked you through 2 new soil-borne issues in California for our strawberry producers. These are fairly recent developments. We're not unique. We in California share these problems with other places in the world and the country, but being the country's #1 strawberry producing state, we're quite concerned about these new problems caused by macrophomina and fusarium. Hope I made my case to you. A precise field diagnosis is not possible. So if you're in a different state and you begin to see dieback issues in your strawberries, I encourage you to again locate your friendly neighborhood plant pathologist or someone qualified to help you with diagnosis, because it will take laboratory testing to confirm what you've got there. And again, the way things happen in our country and our world with movement of materials and such, you likely will see some new disease at some point, maybe not these 2, but something in the future. If they are something you'll see, you'll be likely to be concerned like we are that these are going to be persistent problems that will need to be dealt with in a long-term strategy. And again, really, excuse me, there's really nothing new in terms of the strategies. It'll be similar to what we do for other soil-borne problems, and I went through that with you. That's basically nothing new there, but something that will have to be applied now to new issues, at least for strawberry. Okay, like Tracy before me, I also want to acknowledge and thank a lot of people, especially the strawberry growers in my state of California, and we have a very nice industry group, the California Strawberry Commission, that's funded our research, and then there's lots of people who have helped us with the project, as well as our research team. Really want to acknowledge all those people, and again, really want to thank Kathy and the organizers for allowing me to share this with you today, and with that, I will pass this back to Kathy.
Okay, thank you very much, Steve and Tracy. Are there questions for either of our speakers? While you're thinking about that, thank you for participating today. I want to remind you that the second in the series on emerging pest ID and management will be next Friday afternoon at the same time. We'll have Dr. Robert Martin, who's going to be talking to us about strawberry viruses, and then Mr. Mark Bolda, who's going to be talking to us about management of spotted-winged Drosophila in small fruits. So while we're waiting for questions, I also want to point out to you, oh, the box is not here. Anyway, I have posted the first 2 recordings online, and they are available, and I will be editing last week and this week's webinars hopefully this afternoon and have those posted sometime early next week.
Okay. Oh, here comes somebody.
Okay. Okay, no questions yet. Here is the link for the webinar recordings. Okay, if there are no questions, thanks again, speakers. We really appreciate your participation, and we'll see all of you next week.
Thank you, everybody. Thanks, Kathy.